Bill, and thank you all for staying on time. So we have a lot of time for our Q&A, and I would invite you to just, hold your hand up or go to one of the microphones if you have a question, and maybe what I will do is kick things off. So Stuart, let me start with you, and I was fascinated by this 100-day window that you talked about, and I'm just wondering if you could just comment just a bit more on that, and how you see potential to intervene. I mean, what you're thinking longer term. So, what kind of studies need to be done to actually better, characterize this 100-day window, and why does it close? I mean, I guess what's going on there?
So before I started this study, pediatricians had this idea that kids who were getting asthma and kids who didn't get asthma, it all happened before they learned to walk. So it's around 1 year of age. But I think as time's gone on, there's recognition that very early life things, prenatal exposures, early life exposures, really matter. Mode of delivery, caesarean versus vaginal delivery, early life antibiotics, the family, the dogs, all of these things really seem to impact risk of asthma. So yeah, we had the samples from 3 months and 1 year. That were the samples we had through the child study protocol, and really all the differences we saw, it was strong at 3 months and then not really there at 1 year. And I think what we feel is likely happening is that the microbiome influences immune development, and that's not novel, but and there's lots of data in mice showing that the types of bacteria that you have within the gut microbiome are important for shaping immune responses. Certain bacteria protect against so-called pro-allergic Th2 responses. So I think it's not surprising that it's early. For me, it was pretty surprising that it was so early in that first 3 months. Is where we saw it, and I think it closes because the immune development occurs. Those immune cells are out in the periphery, and at some level the scene's set. I can't tell you exactly when it closed. I can tell you we saw it 3 months. So studies would need to go and look, at more time points to really validate it and prove that. And I think we also need to look in larger samples in different cohorts in different communities to see if really is these four bacterial families that we saw were indicators or biomarkers within projects.
It'd be fascinating to look in the developed world and the non-developed world.
Yeah, so we're excited about that. We have some friends who have a cohort in Ecuador, so quite rural Ecuadorian population. So yeah, we're excited. We don't have an answer.
No, thank you. So, a couple of questions right here.
Just following up on your question, the question for Stuart and Deborah, especially as you are at the same table now. Is there any research, part of your genomic research, that looks at the effect of natural birth versus caesarean one? Because it seems these two things really connect very well.
Yeah, so absolutely. Mode of delivery is related to asthma risk. So caesarean section delivery seems to be associated with increased risk of asthma development in the children, as does early life exposure to antibiotics, whether it's during delivery, antibiotics administered to the mother, or in those first few months of life, antibiotic courses and more antibiotics early on is related to risk. And that's also been related to differences in gut microbiome in the children. Because is no longer.
So how much is also looked into if the child picks up this vaginal microbiota during the natural birth versus caesarean? Is that part of that picture?
I'm certain it's part of the picture, but I don't specifically know the answer. That's better.
So I think the short answer is we don't entirely know how the vaginal microbiome sort of sets the infant's early core microbiome. And there is some work on that. We have to be very careful because the peripartum world is not as clean as one might imagine it to be, where you have these individuals with intact, perfect vaginal microbiome having natural birth, and then people having caesarean sections without any microbial interference. So you have to realize the association with caesarean section is also an association with higher use of broad-spectrum antibiotics, and in some cases a fairly long exposure to the vaginal microbiome prior to the caesarean section in the context of labor. So we have to tease all of those features out to try and understand the relationship. I think to say that there is a relationship between maternal and infant microbiome, probably most people will accept that, but exactly how it is and how that's being set or positively or negatively affected hasn't been entirely certain.
Does the genome of the individual affect the microbiome of the area you're looking at?
Yes. I know lots of data for mice knockout in various immune genes, toll receptor 5, for example. Those mice have different gut microbiomes. So I think absolutely there's interaction between genome and gut microbiome. It's complicated to tease out clearly.
Although I think what we were a tiny bit surprised with, and although our cohorts aren't huge, we're still looking at, in both our non-pregnant and pregnant healthy group of over 300 individuals, diverse ethnicity, we tried very hard to recruit, and different parts of Canada. And with that, we saw actually very stable microbiomes. So although obviously host genome will have some influence, in health there seems to be a fairly classic core microbiome that is common with most individuals, at least for our niche.
And can you potentially treat those at-risk women with your better bacteria to help their pregnancies?
So that would be the holy grail.
Okay.
Sadly, trials both using classic broader spectrum antibiotics and trials using probiotics, some of our colleagues in Toronto have been leading those, have not shifted the microbiome as neatly and as effectively as we would like. So clearly that's what we've got to get to, but the pathway to get there is not simple. And it may be related to the nature of the probiotics and to the fact that women with dysbiosis, that is actually a state that's quite unfortunately stable in them. So it's hard to, you can shift them temporarily, but they tend to revert back to the dysbiosis.
Maybe it's worth saying in this asthma allergy field, probiotics early in life for children haven't been, there's been variable results, but when we put them together in meta-analysis, probiotics have not been effective in preventing asthma. They have prevented other things that in that allergic spectrum, say atopic dermatitis or eczema, there is some benefit there. So I think it's complicated, but again, our holy grail may be the same, is primary prevention of asthma.
And if I just may carry on in the probiotic theme for a minute, I do think that we just don't have the right probiotics yet. There's been industry going, basically anything that's a live microorganism is a probiotic, but we really haven't been very successful at targeting the ideal mix of microorganisms for the area that we're trying to affect. And microorganisms that actually attach the gut or the vaginal mucosa appropriately, establish appropriately. We haven't been using these targeted microbiomes today.
Fiona?
And that nicely leads into my question. So I'm very enthusiastic about microbiome research. It's really showing the huge impact microbiome has, but I'm concerned, as I'm sure you guys are, by this development of probiotics as sort of like snake oil right now, it's a real concern. And I wanted to hear maybe from all of you about comments about how we should move forward to make sure that this research does move forward effectively to these kind of therapeutics that are, and what kinds of experiments should we be doing? Like, for example, with the protective dog, I mean, should we be looking at the microbiomes of these children and their dogs and the environment and see if we could find these specific species? I would love to hear from you how we can address this issue of really coming up with some decent probiotics that are going to be truly therapeutic based on scientific evidence.
Well, I just want to point out that there's a long history of trying to inoculate various environments, not just the human body, with particular microorganisms, and it's been a very unsuccessful history. Basically, these are usually complex ecological systems with a lot of interactions going on that we nowhere near understand. And so, what we do know is that there's a lot of selective pressure that's created a particular community, and when you're trying to work against that, it's not, it may not even be possible. But I guess I'm fairly optimistic that I think a lot of basic microbial ecology is needed to support this. And also, just on the topic of snake oil, I think the important thing is that good scientific research will get us where we want to go. But there will be snake oil salesmen tomorrow, trying to sell things, and people just have to be aware that these are not simple systems, and that there aren't going to be simple answers tomorrow that are commercially available.
And I guess for me, as Deb pointed out, the optimal microbiome or the optimal probiotic supplementation will be very different for the different applications.
Yeah.
So, one size fits all, which is sort of where we are, really I think doesn't work. I'm excited about this early life window. Children are born and then they need to be colonized. And I think that's a niche where we maybe can really influence what happens, because I think as people have said, once things are established, it's really quite difficult to move. But I think in this sort of early life pediatric phase, the rules may be a little different.
Right.
One of the things that we're exploring in forestry is opportunities to adapt to do adaptive management, and one of the proactive options is to assist the migration of tree species outside of their existing range. And we've begun to do this with western larch. The climate models and the tests show that larch can grow well further north than its existing contemporary range. Given time, it's likely that species would migrate north, like all the species present here in BC have migrated north following deglaciation. So the question for you is, and your research perhaps can inform us, is should we also be considering moving the microbiomes along with the trees, but keeping in mind that the trees that we're seeing outside here are probably from Eurasia or Eastern Canada, and they do quite well without their accompanying biomes coming with them.
I think the answer is yes, we definitely should be considering that. First, I just want to point out that trees are going to have to migrate much faster than they ever had to before. So there's been no natural or previous period of history when the climate changed so rapidly. So trees are going to have to move faster. And so I think anything that might assist them is important. Trees in particular have very important associations with ectomycorrhizal fungi that are intimately associated with their roots. So clearly that's an example of an organism that really has to be there for these trees to be, I wouldn't even say optimally efficient, but to survive. I think the other point is the trees around town are not in a forest competing with all of the predators, the insects and so on. So I think that in a forest system, there's probably more of an imperative to have a functional microbiome associated with the tree roots So this is an area that very much interests me and I think is important.
So let me sneak a question in, and Bill, maybe I'll start with you, and others can comment also. So one of the things that has been discussed a lot in just kind of the community, the microbiome community in general, is development of standards, and what's going to be necessary to develop the necessary standards to do comparative studies on to find out what a normal microbiome is in one patient. Forget a population. So can you just kind of tell us what, I mean, I'm asking the question.
No, I can't tell you.
But what I'd like you to comment on is two things. I'd love for you to give us some direction on what we could do as a funder to help move this in the right direction. Because sometimes, people are trying to connect, a cause and effect, and they're doing that prematurely before the standards are developed to allow them to do that. So can you give us some direction on what we might do? And just kind of where this is moving.
Yeah, I think standardization would be great. It would make meta-analyses of different studies much more practical. On the other hand, I'm not that optimistic about a great deal of standardization. There's so much change. Techniques are just things become obsolete within years, a few years. And it's important for people to try different things. Also, we're at a fairly early stage in this type of research, and I think it's important that different researchers are doing different things. Having said that, I think maybe some things that could be done. One would be if you can get more people to analyze the same data. That could be helpful. And I guess another point is that more data will help. It's a two-edged sword because more data is a greater challenge. But the true interactions or correlations will start to stand out the more data you get. And then my last point is I think we start by we look at the most robust relationships that are going to be robust to some differences in methodologies and that's I think where we are today. So, no, I can't help much.
Deb, Stuart, would you like to comment on this? So, Deb, maybe let me ask it this way. So with the data, so vaginal birth versus C-section, for instance, and what we know about kind of that related to the speculation around the microbiome, how do you advise your patients based on that? So when you have an expectant mother, what advice do you give them? Does this factor into your doctor-patient relationship?
To be honest, not yet. I think it's really early stages, and I wear two quite distinct hats, which is, as a researcher, we're really working to try and understand this area, but we're not ready to translate it into the real world yet. The number of factors that influence decision-making around mode of delivery are really trump microbiome decisions for the most part, which is usually related to, is this baby going to fit through? Is this baby having fetal distress? Is there, there's other much more dominant factors. That being said, one of the things that we are urgently looking at is the proportion of antibiotics used in the context of the pericardial setting. And without even studying it, we know that that's definitely having an influence. And yet the counter to that is either treating infection or preventing infection is still a paramount outcome. So we're really in a quandary there, where we'd like to take away a broad spectrum antibiotics, but we need them for survival. So it's difficult.
And Stuart, maybe ask you. So with the a pet, a dog, let's say, and that helping to bridge a child's decrease a child's risk of asthma. Is that something that factors into your doctor-patient relationship, or what is it going to take to get it there?
Yeah, it's interesting. My doctor-patient relationship relies a lot on trying to make people not feel guilty for things they couldn't control in the way they brought their children into the world or the way they've looked after them. Because I tell you, I feel anxious that it's generally mothers blame themselves for mode of delivery, challenges with breastfeeding, thing. So that's my big piece. But I think I think we recognise that there are I think we recognise that the gut microbiome is likely important for a variety of health outcomes, and maybe there are simple things we can do, to modulate that. So we've talked about probiotics, which is quite a significant intervention, but looking at things like breastfeeding, and does breastfeeding mitigate any of those things? Clearly, breastfeeding provides nutrition, but also provides skin contact that can shape the microbiome. So for me, I think some of those important health messages can come through in a nice way. But I think it's important not to make the mums feel bad.
Other questions? We still Peter.
Yeah, I'm just wondering about the intervention in the early stages, as Stuart was talking about. Fecal transplants in later stages, things like inflammatory bowel disease, seem to be quite effective. Why do you think there is a difference there?
So I guess I'm not sure there is a difference. Maybe I didn't understand the question.
Well, if fecal transplants for disorders like inflammatory bowel disease seem to be effective, at least from the clinical trial results one sees. Can't one do similar things in children with asthma, or is that just, when you're saying it's too late, what's the...
Yeah, I wonder if it's, well, I don't know if it's too late, but I think it's harder to change things once they're set up. I think in some of these, I think we're looking at subtle dysbiosis in that these children who have asthma and allergies are otherwise generally quite healthy children. And I think they're not on the same spectrum of people who maybe have inflammatory bowel disease or C. diff infection, where the skewing is much more dramatic. So I think we're looking at those people. Why I sort of hesitate to sort of be advocating intervention is I think there will be a lot of ethical and logistic challenges having interventions, even probiotic interventions, in a child who's 1 to 2 months of age. I think our standard to prove that is safe is going to be very high, and that standard is different, likely different ethically to an adult patient who's suffered, say, with C. diff for a very long time, where they're consenting for that. So that's really what I'm flagging, I think, is just the challenges of translation.
Can I just add to that? We've got an interesting practice that I just found out about where some of our midwifery colleagues have taken this information, which we're talking about today, and have begun advocating for their women who end up with a caesarean section to do vaginal swabs, which they're then inoculating the mouth of these infants. And at first pass, you think, okay, I kind of get where we're going here. But it's back to some of the things, Fiona, you talk about over and over. We have to do the basic studies. We have to establish what the microbiome is in the normal situations and the various subtleties of abnormal through to the extreme. And every intervention, be it something as minor, it may appear as that, is they may actually be inoculating a highly pathogenic organism that may be part of the microbiome of the woman under the circumstances of that caesarean section. So if we take group B strep or pathogenic E. coli and inoculate a newborn with that, perhaps that's not the wisest thing to do. So we really have to go back to basics. And Brad, I think you were alluding to that, is that I think we unfortunately have to do what may appear to be very boring science, the ordinary experiments of this is health, this is one intervention, how does it shift? The next one, how does that shift? And until we understand this complexity a bit better.
I just want to point out that the applications of this research are not limited to inoculating people with whole organisms.
Yes.
So, these effects are all based on mechanisms, and when we tease out the mechanisms, we may find much more acceptable and simple ways to get the desired effect. There may be just particular molecules on the surface of these bacteria that need to be present at a certain place at a certain time. So that there's, again, that points to the need to do the research and to do it thoroughly.
Rob, I was just going to make a point. It seems that it's going to take a little while to get the probiotic system worked out. Is there more we can do on sort of narrow spectrum antibiotics in the interim? And you know, the broad spectrum antibiotics seem to be a major factor in adjusting or changing our microbiome. And I wonder if that's an area that we might want to, as a group, because one of the things that Sally addressed was a very important matter to take care of.
I think you're totally on the right track there. You know, I think we've gone broader with, you know, one drug kills everything kind of approach, and there are some situations in medicine where that's helpful. But I think, in fact, I think we need to start going in reverse, which is subtle changes in the microbiome may be very, very important, and really, really narrow, targeted antimicrobials for a very specific reason that is almost very tailored to drugs, getting almost to the pharmacogenomics kind of notion for each and every problem may be much better. The challenge is industry doesn't want to develop many niche drugs. They want the big, you know, fabulous drug that will kill everything, and so we've got to work on that.
Yes.
Just out of blue question, it just came to my mind, is anyone in the health looking at the issue of sexuality and how much people through different sexual practices, although they exchange sexually transmitted diseases, but how much the microbiome play a role there, and can be kind of also tracked as a life experiment kind of thing, that some people are healthy in certain situations because of microbiome or not. Is anyone looking into that?
I'm not aware of it. I think there's plenty of studies showing that you don't have to be that intimate. You know, just looking at how families tend to have more similar microbiomes, just being in proximity to people clearly does shape your microbiome. So I think, you know, the answer to that would be there would be an effect, probably.
So we are doing that in a small way. Our collaborator, Kieran O'Doherty, is a social scientist working with us, and one of the things he's been doing is a very, very detailed series of studies from surveys down to focus groups to really understand all of the different behaviors that individuals are involved with, from sexual practices to use of cleansing products, douching, various things, the number of partners, the diversity of partners, and trying to understand how that shapes, at least in this case, the vaginal microbiome. And some of our classic microbiology studies have suggested that there is a significant difference related to who your partners are, numbers of partners that you have, and the nature of practices, and that it does change. That being said, it doesn't necessarily, outside of the STI arena, necessarily translate into practical advice, but at least it's beginning to understand how behavior shifts the microbiome.
Phil, can I just ask you a question so most of these antibiotics are used in farm animals, and so has anybody looked at the microbiome of farms? I mean, where these animals, where their excrement's being scattered and whatnot, to really look at shifts. And I'm really thinking even the forests that broach those labs. I mean, has anybody actually done that kind of a study yet to see what kind of effects those antibiotics are having in the field?
I'm not aware of cases where people have studied whether such, you know, intensive use of antibiotics is actually shaping entire environments. You know, most of the studies have focused on the transmission of resistance and so on, which clearly goes on within the surrounding environments. But
sounds like a good out for you.
Yeah.
Any other questions? Well, with that, please join me in thanking Bill